neurotransmitters. How? So now we are moving on into neurotransmitters, which is another kind of chemical signaling molecules. So when we talk about neurotransmitters, these are synthesized in the neuron, right? And they are stored in thin walled sacs, which we call it as synaptic vesicles.
Yeah.
So, so if this is the neuron, so this is postsynaptic neuron and this is presynaptic neuron. You have these neurotransmitters as being stored in a vesicle, which we call it as synaptic vesicle. So what happens is this vesicle move towards the end of the membrane and they form a invagination to release these neurotransmitters. So this invagination is called as synaptic cleft.
Yeah.
So this happens when there is an action potential, which means when the neurons are getting transmitted from one to another, or when there is a electrical signal being passed from one to another. So this is postsynaptic neuron. This is presynaptic neuron. So using these release of neurotransmitters, so you have receptors that are here. If the neurotransmitters are specific to the receptors, they bind and they get passed on to the next neuron, and the process proceeds. So these are messages that help us to sense, feel, do any action. Now, when we talk about the types of neurotransmitters, they are peptides, amines, nitrous oxide. These are the different kinds of neurotransmitters. When I talk about peptides, peptides are proteins, correct?
Yeah.
Some kind of protein. So common example what we take for peptide is neuropeptide. Y. Neuropeptide Y. So this is responsible for example, you have a feeling of eating when you see a food, right? All
Yeah.
that is because of this neurotransmitter, neuropeptide being synthesized as a result of you looking at the food. And the next one is amino acids, which is amines. So amino acids, common example, we take GABA. I think in the last class itself, I mentioned about this gamma aminobutyric acid.
Yeah.
Gamma aminobutyric acid, glycine, glutamate, all these belong to neurotransmitter classes. They play a very important role in fast synaptic transmissions.
Yeah.
So these are amines. So you also have dopamine. Have you heard about dopamine anywhere?
Yeah. So emotions controlling, right?
Yes. So in the brain, so for the movement and regulation in the brain, then noradrenaline, serotonin for mood swings, and your flight and fight response will all belong to these amine category of neurotransmitters. And nitrous oxide, it is a modulator of neuronal function. So all your neuronal functions are modified or modeled and using this as what's that? Nitrous oxide. So these are some examples or types of neurotransmitters. So already how these neurotransmitters act is what we have seen now, right? So they just present in the vesicle, they come to the surface, they get invaginated and they release and bind to the receptors. That is how they mediate their function. This is what we call it as mechanism of action of neurotransmitters. Similarly, we have finished hormones, we have finished neurotransmitters. The next one which comes into play is the cytokines, which is also a signal molecule. So when I talk about cytokines, so they also have some receptors on the target cell. And so they bind to the receptors on the target cell and they catalyze a set or series of reaction. So what are the different kinds of cytokines that we have? So cytokines or on what and all do they act? So cytokines can act on a lymphocyte, which is a WBC, right? So they
Yeah.
can act on macrophages, endothelial cells, which is present in skin and all those things, and fibroblast, So these are their target organs. So cytokines can only act on such target organs. So when there is a stimulus, the cytokine producing cell will release the cytokine. So the cytokines will bind to the receptor which is present on the target cell. It can be any of this target cell, right? And so it binds to the receptors and it catalyzes the series or set of reaction. Okay. Next coming to mechanism, next coming to calcium ions.
Sorry, 1 second. One question.
Mm.
Are these cytokines released during apoptosis, or like just how when the cell is attacked, or
By
when?
a stimulus, any kind of stimulus.
Okay.
Any, for example, when like how hormones are released when you have a mood swing, how hormones are released when there is a menstruation period for a girl. Same way, when there is an external stimulus, right? And at
Yeah.
that time, and you have these cytokines ions being produced. So it is mostly when you're sick. For example, you're sick in
Yeah.
response to any kind of infections, disease. So that is the stimulus here.
Okay.
This an infection is a stimulus here. So mainly for the purpose of producing immune response, you have the cytokines.
Yeah.
Okay. Next one
Okay.
is calcium, which is also a chemical signaling molecule. So this calcium, which I am talking about, Ca2 plus, it acts as the second messenger. I remember taking this right. So there are
Yeah.
two.
So it doesn't
Uh,
act like on its own, and
yes.
it uses something else.
Yes, so it is in the signal transduction pathway. So calcium doesn't is a not a first messenger. It is produced in response to something another chemical, another signaling molecule, right? And so so this type of calcium response happens, and when you contract your muscle, and then when period of fertilization, right? So at that period only, this second messenger production, which is calcium, takes place. So high level of this cytoplasmic calcium may lead to a cell to undergo apoptosis. What is apoptosis?
Cell death. Yeah.
Yes. So when there is more concentration of calcium, then there may be apoptosis happening.
How does this happen?
How does it happen in the sense?
Uh. Hmm.
When the concentration of when the concentration of calcium is more in the cytoplasm, it can cause the cell to lead to its own death. So high
Okay.
level of calcium is not encouraged or is not advantageous.
Yeah. Okay.
Apart from that, it has other roles. But this high level can lead a cell to program cell death.
Okay. Okay. Yeah.
Calcium plays very important role in enzyme activity. Then it is helpful in regulating any ionic channels for plasma membrane transport along the plasma membrane transport, then active transport, activity of ion bonds. And it also plays an important role as a cytoskeleton. You'd have heard in some organism calcium carbonate as a cytoskeleton. So all that can be considered as a function and right, but high level of calcium may lead to apoptosis, which is programmed cell death.
All right.
Fine. And
Yeah.
so we have now spoken about all the different kinds of chemical molecules all the different kinds of molecules that lead to signaling, like hormones, neurotransmitters, cytokinins, and calcium. Its type, and each one of them have their own kind of action or activity to take place when there is a stimulus, right?
Yeah.
And so now these signaling molecules which are produced, it can be anything, whether it is a hormone or a neurotransmitter or whatever it may be. Either these molecules are produced in the same area where the target cell is present, or in an area away from the target cell. They can be produced either near to them, or they can be produced
Away.
farther away from them. So, depending upon the distance of secretion, okay, depending upon where these molecules are synthesized, either near or farther away from your target cell. And
Yeah.
you have two different types of signaling. One is autocrine signaling. Auto
Hmm.
means what?
Self.
Yes. So self. So the cells secrete signaling molecules. So this is the target cell. This is the secretion cell. The cells secrete the new whatever hormones in their own cells and act in the same cell. You're getting what I'm trying to say?
Yeah. So it's
So
produced in the cell itself and then acts in the cell.
yes, yes. So they bind to the receptor that is present on the membrane and they catalyze the reaction,
Yeah, got it.
whatever reaction. For example, cell differentiation. So a cell has to differentiate. So
Huh.
the hormone of cell differentiation is produced in the same cell and it gets differentiated in the same cell. You're getting an understanding?
Got it.
So example is cell differentiation. On the other hand, this is autocrine. So self-secretion, self-action. On the other hand, you have another kind of signaling, which is paracrine signaling. Autocrine, paracrine. So in this case, the signaling molecules are synthesized somewhere and they have to travel to reach the target cell. So the distance can be farther away, but it's two different cells. So secretion happens in one cell and the target cell on which it acts upon is another cell. It can
Yeah.
be in close proximity or distant proximity.
Okay. Yeah.
Okay. So the signals gets diffused either by the process of endocytosis or exocytosis, and they
Hmm.
move outside and bind to the receptors of the target cell and they catalyze the reaction. For
Okay.
example, the contraction of the muscle. You
Hmm.
have the hormones synthesized, right?
Yeah.
So it is synthesized, it has to reach the muscle and it
Hmm.
has to make you contract. This is a second type. So autocrine, paracrine. The next one is endocrine.
Hmm.
So this is close proximity, I said. Here, secretion happens in one cell and it has to reach a long distance
Okay.
for a target cell.
Yeah.
Most of the hormones will come under
Hmm.
the category of endocrine.
Yeah.
Hormones are synthesized somewhere, but the action is somewhere.
Hmm.
You're getting an understanding?
Yeah.
Autocrine, paracrine, and endocrine. Is it okay? The
Yeah.
next signaling is juxtacrine. Any idea what it is?
Just the position is opposing, so something like that.
It is contact dependent,
Oh, okay.
which means the cells has to be in physical contact with one another.
Uh-huh.
The signal molecules, no, they are not free. They are bound to the surface of the signaling cell like this.
Ah, okay.
Okay, and they have to bind to the receptor. So which means two cells has to be in contact with each other.
Hmm. Okay,
Okay,
got it.
so for here, in this case, as you have ions and all passing through gap junctions, synaptic cleft between the two
Hmm.
neurons, creating a small space. So all this can be taken as this
Yeah,
juxtacrine
but synaptic
uh,
in the synaptic cleft, isn't there, like space between them?
synaptic cleft.
Isn't there like space between the two neurons?
Yeah, there is. It comes under paracrine. It
No,
may
yeah, yeah.
be very close
Yeah, that's what
in
I'm saying.
contact. Okay,
Yeah.
so there may be gap junctions. That is
Hmm.
also possible. So when they are contact dependent, when then the signaling molecules are not freely released from the secreting cell. You call that
Yeah.
as juxtacrine.
All right.
Is it fine?
Okay.
So try to remember autocrine, paracrine, endocrine, and juxtacrine. These are the different kinds of signaling methods based upon where they are secreted and where they travel to act.
Hmm.
Is it fine?
Yeah. All right.
Okay. So now we are talking about receptors. Where are these receptors present?
On the surface of the cell.
Yes, so mostly these receptors are present on the transmembrane. So we call
Hmm.
it as transmembrane receptors in the plasma membrane, right? So
Yeah.
the ligands which we are talking about, that
Hmm.
whatever which gets attached to the receptors, they cannot cross the cell membrane. So what happens? These ligands bind to the receptors that is present in the plasma membrane. And in the
Yeah.
beginning itself, we told they are very specific to each other. Not everything can bind with everything, right?
Hmm.
So this receptor, ligand and the receptor binding, they produce the response by activating the proteins that is present. So that proteins are called as second messengers. That's
Yeah.
what we
Oh,
have
yeah.
been learning, correct?
Yeah.
Right,
Yeah.
which can be calcium or any other protein molecules.
Yeah.
So how do they regulate these responses? They
Using
usually
nitrous oxide.
regulate. Yes, tell me.
Using nitrous oxide.
No, that is one type. Generally, I'm
Okay.
talking.
Got
Nitrous
it.
oxide is one method, one molecule, one signaling molecule, like a hormone neurotransmitters. It is also one signaling molecule. So
Yeah. Okay.
mostly they activate enzyme.
Hmm.
So how do they cause the responses? It is activation of enzyme by phosphorylation, which is addition of phosphate.
Yeah. Phosphorylation. Yeah.
Okay, and then
Phosphorylation
uh,
also done in like some other process, right?
what can be done? Hmm,
Okay. Yeah. I
hmm.
said
Yeah, yeah. Tell me.
no.
No,
No.
basically, when a signaling molecule is released from the cell, they bind to the receptor. Either they activate a response, or
Hmm.
they if they are not if they if as we are talking about ligands, the ligands cannot directly come inside the nucleus. So
Yeah.
they take the help of the second messengers, right? So they
Hmm.
attach to the receptor, they produce a second messenger, and the second messenger will go into the nucleus and the process starts, correct?
Yeah.
That's how it happens. So how does this basically mm, there is a series of reaction that is happening. Like you have some enzymes. It is all enzymatic metabolic reactions only, no? So it is
Yeah.
involvement of enzyme. Mostly the enzyme undergo phosphorylation.
Yeah.
So once they undergo phosphorylation, they then enter into the nucleus through the pores of the nucleus, and they
Hmm.
finally cause transcription and whatever the gene
Gene
expression which we
expression.
are talking about. So, right. So there are two different types of receptors which we have. This is the common way of the receptor and the how they catalyze a reaction or how they initiate a response. So there are two different types of receptors. One is cell surface receptors, which we are already talking about, right?
Hmm.
Cell surface receptors is one that is present on the surface of the
Yeah.
membrane. Next one is intracellular receptor. What is intracellular receptor?
It's inside the cell. So that's used
Hmm.
for like autocrine, right?
Pardon me? Intracellular receptors means autocrine. I don't get you.
No, I'm saying the intracellular is like inside the cell. No.
Yes, so they are present. Correct, very good. So they are
Yeah.
present inside the cell, located inside the cell. So they are present in the cytoplasm or sometimes in the nucleus of the cell also. Right. Right. So the cell surface receptors that are present on the plasma membrane. So if it is in the plasma membrane, the ligand binds over here and it catalyzes a series of reaction. Correct.
Yeah.
And so it may be a hormone, neurotransmitter, cytokines, or whatever it may be. But in case of intracellular receptors, where your receptors are present inside, and what happens is they bind to small lipid derived signaling molecules. For example, steroid hormones. They do not bind to everything. You're getting it? Since
Yeah.
they are hydrophobic in nature, they bind to this small lipid molecules. For example, I'm telling steroid hormones, and they cross the plasma membrane, and that's how that's how the neurotransmitter gets its way into the receptor. You're getting
Yeah.
an understanding? If
Yeah.
this is the if for example there is a hormone here, or like what to say, this can directly bind to the receptor and move inside. But one which is
Mhm.
present outside, and since the receptors are present inside and they cannot cross the membrane, what
That's to
happens is
bind
they has
to
to
a
bind to
non-polar.
something else.
Yeah.
Yes, which is able to pass into it. Along with it, they bind to the receptor inside.
Yeah.
So this, at that time, we call that as hormone receptor complex. Hormone receptor. This complex can directly move into the nucleus and cause whatever reaction it needs to happen.
Is that a temporary complex, right?
Yeah, it should be a temporary complex because it catalyzes a series of reactions and it has to get separated.
All right.
So it has three regions. When we are talking about the cell surface receptor, it has three different regions. And one is extracellular ligand binding domain.
Okay.
Okay, for example, if I have the plasma membrane, let me consider this as a plasma membrane. So phospholipid bilayer, right? So
Yeah.
just with my diagram. So this is the thing. So so this is the receptor protein which we are talking about. So it
Mm-hmm.
has a extracellular outside, it has a ligand binding domain.
Yeah.
And towards the inside, it has an intracellular ligand binding domain. This is extracellular.
Mm.
This is intracellular.
Yeah.
So it has an extra outside one domain, binding domain, and inside one intracellular domain. And in between this one, no, it has a transmembrane hydrophobic domain.
Yeah.
Transmembrane
Wait,
hydrophobic domain because it is a lipid
okay. Yeah.
tail.
Yeah.
So these are the three regions of a cell surface receptor.
Mm.
Three parts or three components of a cell surface receptor. Is it fine?
Yeah.
So this is about the first type of receptor. What is the second type of receptor that we are discussing here?
The extracellular.
No, extracellular or no, one is cell surface receptor. The
Yeah.
second type is
Yeah. What? Which one?
no, first there are two types of
Yeah.
receptors. One is cell surface,
Also.
another one
Ah.
intracellular,
Ah. Yeah. Yeah. Yeah.
intracellular receptors. So these can react or interact with ligands that can cross the cell membrane because your receptors are present inside, correct?
Yeah.
And so mainly we take the examples of hormones only. So here again, these intracellular receptors are of type 1 and type 2. So this type 1 is present in the cytoplasm. So I can call them as cytoplasmic receptors.
Okay.
And then only they move into the nucleus. For example, you can take progesterone, glucocorticoid receptors, all this comes under type 1. On the other hand, the type 2 is
Gluco what?
direct glucocorticoid. All these are hormones. Progesterone, androgen, glucocorticoid receptors.
Okay.
Type 2 is directly present inside the nucleus. These are called as nuclear receptors. This is cytoplasmic receptors. This is nuclear receptors. So they are directly involved in transcription. They do not need any enzymatic, like that first we saw, no, there should be some phosphorylation happening. That
Yeah.
and all
Yeah.
is required here. They directly bind to the receptor present in the nucleus and they catalyze the transcription and by binding to the domain. And so for example, this you can take thyroid receptors,
Hmm.
your retinoid receptors in your eye. Even insulin belongs to this category.
Okay.
Here you can, if you want to easily remember, you can remember progesterone, androgen, glucocorticoid is also easy, but easy to be more. Here,
Hmm.
thyroid insulin you can remember for type 2. So this is cytoplasm, this is nucleus.
Hmm.
So the insulin receptor in specific is little more explained here. So the insulin receptors, we know insulin is a large protein,
Yeah.
right? So they are permanently attached to the cell membrane.
Okay.
The protein I'm talking about. So they are
Hmm.
permanently attached to a cell membrane and they have multi-domain structure. So they have both extracellular and intracellular domains.
Yeah.
Like, for example, I'll just show you, just try to draw. So if you take this, let me assume this as the plasma membrane. And so if you assume this as a plasma membrane, and if you see the insulin, I think it will come like this. Somehow it will be. Why did I call it as multi-domain is, see the structure it has, it's not simple. Did you understand why is it called as multi-domain?
Yeah.
See the domains, this one, this one, this one, this one, this one, same
Mm.
ratio. Right, so
Yeah.
that's why it is called as a multi-domain. It's a very large protein and it is a multi-domain structure where
Yeah.
you can see this is the extracellular domain outside the plasma membrane. This is the intracellular domain.
Mm.
So this extracellular domain, which means the outside one, right? They
Mm.
bind to the insulin and they activate an enzyme called as tyrosine kinase. This is what I call it as insulin receptors.
Okay.
So this belongs to what type?
The
Which
intracellular.
type of signaling?
Wait. This is because it's gonna be endocrine, no?
Yes, correct.
Yeah.
Okay, so when you see the detailed explanation is not essential, just you can remember it's a multi-domain structure. It has an extracellular domain, intracellular domain, and only the extracellular domain binds to the insulin protein and which because this is a receptor. So this is an insulin receptor we are talking about. This is the structure of an insulin receptor. Okay. So
Mm.
this region only will bind to the protein to catalyze the production. How will it catalyze the production? By activating this enzyme, which is called as tyrosine kinase.
Mm.
Is it okay?
Yeah.
Any clarifications you need?
No, it's fine.
Okay. So this is about intracellular protein. Now coming back to the cell surface, what is it? Cell surface receptors. So intracellular receptors, we have seen what are they, some examples, what are the different types of intracellular, type 1, type 2, we have seen. Coming to cell surface receptors, they are also of different categories, mainly three. First one is, I think, enzyme linked.
Sorry,
G
what is this again?
protein,
It's a cell surface receptor.
cell surface receptors.
Okay.
There are two types of receptors, right? One is cell
Yeah.
surface, another one is intracellular.
Mm.
Intracellular receptors are of two types, which is type 1 and type 2. Type 1
Yeah.
is present in the cytoplasm, type 2 is present in the nucleus, where we saw the
Yes.
example of an insulin, correct?
Yeah.
Like now we are moving on to cell surface receptors, which is also of three types. One is enzyme linked receptors. Second one is G protein linked receptors. And the third one is ion channel linked receptors. Ion channel linked receptors. So these are the three different types of receptors. Is it okay?
Yeah.
So we saw about cell surface. So on the surface we saw, inside the cell we saw, there is one more thing which is left behind. What is it?
Not
Surface
sure.
we saw. So if you think to be this is the membrane. On the surface we saw, on the inside of the nucleus we saw, that is cytoplasm and nucleus. So what is that is in between?
The membrane?
Transmembrane receptors.
Oh yeah.
Okay, so which is in between. Okay, so these transmembrane receptors are made up of hydrophobic amino acids. They are made
Yeah.
up of hydrophobic amino acids. Like, do you know the name of hydrophobic amino acids?
Mm.
No? Okay.
Uh,
Phenylalanine, leucine, tyrosine, all
yeah.
this comes under the category of hydrophobic and amino acids.
Okay.
Then talking about where is this hydrophilic amino acids like serine, asparagine, they are
Hmm.
located on both the side of the membrane.
Okay.
Hydrophobic is present in the transmembrane, that is in between, but
Hmm.
hydrophilic is present on either ends. So
Hmm.
these hydrophobic signaling molecules or signaling chemicals mainly include these steroids. Why is it steroids very specifically coming? If it is if it in the hydrophobic, most of the signaling molecules are
Steroids are
hydrophobic.
humans, no?
Uh, because they can cross through the membrane due to the
Yeah.
hydrophobic nature and bind to the intracellular receptors
Yeah.
and catalyze their reaction.
Hmm.
On the other hand, the hydrophilic molecules like cytokines, neurotransmitters, and all
Yeah.
cannot penetrate to the membrane due to their hydrophilic in nature. So they have to use either transmembrane protein or cell surface membrane protein to
Yeah.
catalyze their reaction.
Hmm.
Is it okay?
Okay.
So these are the three different types of um, cell receptors.
Hmm.
So we have spoken about the different types of signaling molecules. We have spoken about the different types of receptors. As we have spoken about how each of the signaling molecules function, what are the different types. Now only we are going into the action, which is signal transduction pathway. Signal transduction path. You are an SL student only, no? Sorry, HL student only, right?
Yeah. Yeah. Yeah.
Signal transduction pathway. So transduction means what? What is transduction? Generally, English term or whatever. Transduction means what?
It's like across something like like changing your form or something.
Transferring, changing, transforming. That's what we call it as transduction.
Yeah.
So what is happening here? When a molecule is released, they bind to another molecule, and they cause change, right? And they're trying to cause change in the structure, and they are carrying out a kind of signal or transporting the signal from one to another. That is
Hmm.
from an effector organ to a target organ. Correct? So
Yeah.
same thing in case of whether it is present on the cell surface or whether it is present on inside of the cell. And there are some kind of changes that is happening for the action to take place, right? Right. So that is what we mean here by signal transduction pathway. So there are changes that is happening in a stepwise manner or series. So either we use the term signal transduction or we use signal transduction cascade. So cascade is nothing but a series of biochemical reaction.
Hmm.
So when does this reaction happen? This reaction happens in response to some stimuli. So
Yeah.
when there is a stimuli, you have signaling molecules released, which has
Hmm.
to bind to the receptor either inside or outside, and
Hmm.
it leads to a set of reaction, which I call it as transduction cascade. Okay.
Yeah.
So what are the different steps? Basically, what and all should happen?
So like when a molecule, it binds to a receptor, and then there's a conformational change, and then
First step is what can it be?
a ligand.
No. First step is receptor activation. So
Okay.
the receptor gets activated. Second is signal amplification, meaning
This
amplification means what? What am I?
the signal is amplified, or it's like
Becoming more, or
yeah.
it's just. So receptor activation is the receptor. Where is the receptor present? It can be inside or on the surface of the membrane. So when a
Hmm.
ligand binds to the receptor, it gets activated. And we already spoke about specificity. So specific ligand, specific receptor, and they get activated. Signal amplification is nothing but lesser amount of signal is trying to get amplified to more and more until they reach the target organs for a larger response. That's what we call it as signal amplification. So it may occur through several messengers and several downstream until they reach their target organ. So for example, the hormone is synthesized somewhere. They bind to the cell surface protein of the nearby one, but they travel all along by activating all the enzymes that is present by the process of phosphorylation. So there is a chain reaction that is continuing until it reaches the organ which is had to reach, right? That is
Yeah.
what is called as signal amplification. Second step. Third step is second messenger production. Second messenger production. So common, what is the second messenger? Common second messenger apart from not calcium. Calcium is another signal molecule. What is another common second messenger? I think I told you once. Cyclic
There's
AMP. CAMP. This is the common.
no, we didn't.
No, we didn't do. Okay. So this is the common or most what is it? Second messenger that is being produced.
Okay.
Cyclic adenosine monophosphate.
Okay.
So majorly they play a very important role in cell signaling and cellular function. And why they are called as second messengers? Because they are produced by some receptor. There is already a first messenger which has reached the cell. With that response, this is being produced. This is not directly being produced. It's synthesized in response to another receptor. You're getting?
Yeah.
So I think again you will have this in C2.1, I think, this CAMP. You will have little more of it, of the CAMP molecule. So first is receptor activation, second is signal amplification, third one is production of secondary messenger, and fourth one is activation of protein kinases. So what happens? Activation of enzymes means what should happen here, which is protein kinases. How do they get activated? I told you, right? One reaction. Addition of phosphorylation.
Phosphorylation. Yeah.
So yes, so that is how they get activated. So under normal condition, now whatever they are doing now is in an inactive state. So what happens is once the CMP, that is secondary messenger, is produced, it activates the enzyme by adding a phosphate group to the kinases that is present, any tyrosine kinase, any kinases. That's why we are generally mentioning it as protein kinases. So these are the 4 steps of signal transduction cascade or pathway. Very important extended response question.
Yeah.
Okay.
Good.
And so this is about signal transduction. Let's move to the next part. So whatever we are talking about now is for the purpose of a response, correct? There is a signal molecule produced, they bind to the receptor. So basically, it's like all these reactions are happening, and finally for a what to say, a kind of response to happen. So what is that response? It's commonly gene expression. For example, if I take a signaling molecule, it binds to the receptors, right? Once it binds to the receptor, you have either a secondary messengers. You have it becomes a complex. So once it becomes a complex, then it reaches another cell where you have the production of secondary messengers. So this secondary messengers, what they do, they take part in various roles. So it can be anything. For example, cytoskeleton making, cytoskeleton regulating protein. They form a cytoskeleton. Some form or become a metabolic enzyme. Some take the role of transcriptional factor. So all this metabolic reactions are mostly a faster response, faster cellular response.
Yeah.
And this transcription, that is translation gene expression, it is a slower one.
Oh, yeah.
So all this which I am talking about out is nothing but the signal transduction pathway or signal cascade, right? Now we are talking about response, but somehow or the other this has to end. It cannot keep on going.
Termination, right?
Yes, that is where termination of the signal. So once the signaling molecule has finished their work, so it has to be removed from the system, which you asked me, right? Whether they will, what happens. So again, this is also having some steps. First one is degradation of the signaling molecule. So they get degraded. For example, there are enzymes like acetylcholine esterase. I don't know whether you have heard of these names. Acetylcholine esterase. So this will degrade acetylcholine, which is a neurotransmitter in the synaptic cleft.
So this acetylcholine estase is an enzyme.
Yeah, this is an enzyme. This degrades this neurotransmitter acetylcholine.
Okay.
Okay. So by degrading a neurotransmitter, it can stop the reaction. Second is diffusion of signaling molecule. They move out.
Hmm.
If their action is over, they move out. Third one is reuptake. They have been reabsorbed by the presynaptic neuron using some special transporters. So
Okay.
these are the, then you have inactivation. So for example, one example is, I think, beta adrenergic, I think, adrenergic receptors. If you expose these receptors to adrenaline for a long period of time, they get deactivated.
Oh, okay.
Okay, how is these receptors become phosphorylated by kinase and they are not available for the binding purpose. So by
Hmm.
inactivation, so at least learn one example for each how I'm mentioning. So if degradation means acetylcholine, diffusion you can write anything. Reuptake, I think what example mostly
It's just
serotonin will come.
okay.
Serotonin is an example of reuptake. Like, then here I told you beta adrenergic receptors inactivated by adrenaline. Then you have feedback inhibition. What is feedback inhibition? You know,
Yeah, I think I did inhibition in enzymes. They're similar.
feedback inhibition is the levels control their production. For example,
Yeah.
when insulin becomes more, sorry, when glucose concentration becomes more, insulin is produced to lower them. Am I right or wrong?
Hmm.
You getting it?
Hmm.
When it is becoming less, glucagon is produced, right? That is
Glucagon,
called
yeah.
as feedback inhibition in response
It's
to some supply.
it's
So
when
these
glucagon prediction production inhibiting the insulin production.
glucagon production will not inhibit. And when insulin is more, then glucose production will be more, correct?
Yeah.
So in that case, glucagon has to be produced to make the levels of the glucose low.
Yeah. Okay.
Opposite reactions. That's what we call it as feedback inhibition.
Hmm.
So these are the 5 different ways of termination.
Okay.
So you can use these same examples: degradation, reuptake, inactivation, feedback inhibition, insulin you can use. So this
Hmm.
is how they get terminated, your signal transduction pathways.
Hmm.
The next one is, I think, what else is there? Okay. Since we have spoken about out cell surface receptors and intracellular receptors with examples, transmembrane receptors also some examples we need to know, where
Hmm.
you have transmembrane receptors. Mhm. So in the beginning itself, I mentioned that neurotransmitters are some substances or chemicals that are being stored in the thin wall sacs called as synaptic vesicle, right? So
Yeah.
inside that vesicle, you have thousands of neurotransmitters. It is not one or two. And they
Yeah.
carry the signal from one to another by binding to a receptor that is present either on the membrane or something. So there are three possible outcomes of electrical signals depending upon the neurotransmitter. One is excitatory neurotransmitter. Excitatory neurotransmitter. Outcomes I'm talking about.
Hmm.
Excitatory means what? Excite means what?
Uh, to get active
Excitation.
inside. Yeah.
Uh,
Yeah.
so it is like making the message move forward at a greater speed. When you get excited, you jump, right?
Hmm.
Same way, the level of excitation becomes more here. The neurons get excited and they get transferred as a forward message in a greater speed. For example, your adrenaline, noradrenaline, all comes under the category of excitatory neurotransmitters.
Alright.
Next type of response can be inhibitory
Everything.
neurotransmitters. So in this case, they either block or prevent the excitation.
Hmm.
For example, GABA, the gamma. So this can behave as an inhibitory neurotransmitter. Next one is modulatory. Modulatory means the name itself is saying modulatory
Hmm.
means
Modulating.
yes. What is modulatory means? What do you think? Neuromodulators, what can they do? This is excitatory, this is inhibitory, then what can this be?
This maintains, right? This modulates.
Yes, so it is in between both. It modulates.
Yeah.
It can create both excitation and inhibition depending on what type of receptor they have. So example here, acetylcholine and dopamine. So these are the three different outcomes you can expect, either excite or inhibit, or it can be both in response to them, right? So
Hmm.
when we are talking about the traveling or responses, basically there is something called as membrane potential that is created. What is
Yeah.
membrane potential?
So it's basically a charge across the membrane.
Yes, so it's a difference in the electrical potential between the exterior and the interior membrane. So when they are electrically, like when these are electrically excitable cells, these neurons, right? And
Hmm.
so an action potential is generated when the membrane potential is more or
Hmm.
less. You got it?
Yeah.
So an action potential will be generated when the difference in that, so this is the outer membrane, this is the inner membrane means,
Yeah.
depending upon the charges, you have an action potential being generated. So acetylcholine is the first or the primary neurotransmitters that is associated with motor neurons. I think you would have known about three different types of neurons. You know,
Yeah. Motor,
what are
relay,
they?
and sensory.
So for the last one is the motor neuron. First one is the sensory relay, and next the motor, right? So this
Yeah.
acetylcholine is a primary neurotransmitter that is associated with the motor neuron. And it plays very important role in heartbeat, blood pressure, muscle contraction, your sleeping patterns, and all those things.
Yeah.
And so when there is, so for example, a muscle has to contract. You call that reaction as, excuse me, you call that reaction as neuromuscular junction
Okay.
because the action is taking place in the muscle mediated by a neuron.
Yeah. Yeah.
So the synapse that happens between a neuron and a muscle is called as neuromuscular junction. You're getting it?
Hmm.
Hello?
Yeah. Yeah. Got it.
Okay. So In this neuromuscular junction, there is a series of event. Maybe I'll take you through this in the next class, because again you have a little complicated things. If I start, I cannot finish. I have my next class. Okay, so maybe I'll take you through these series of steps in the neuromuscular junction in the next class.
Okay.